Okay, since we have seen now the Sanger sequencing, it's time to proceed with really NGS sequencing, so next generation sequencing. And what I said in the previous video is that we will focus on Illumina data, and therefore let's just have a look on how Illumina managed to speed up enormously the sequencing process. So imagine that you want to sequence some piece of DNA, which is this one. Then what you do is that you use a two ligand model here. So you have this green piece of DNA and this purple piece of DNA, and you have this thing here, this glass, whatever it is, I don't know what material it is, that you have fixed on the top of that this red, purple and green pieces of DNA. So if your DNA has a purple piece here, it will hybridize with the green one and vice versa. Purple and green are complementary. Okay, then what's happening is that DNA is bending here. I don't know how this is happening, to be honest. Maybe it's just a matter of physics here, that it bends, and this end then can hybridize here, so you'll get this small bridge. So this is a single molecule bridge. And when this happens, you put some primer, and then you just start to synthesize complementary, reverse complementary molecule here. So like that. But when you finish this process, when you sequence this one, you will have two pieces. And then again, this will bend over, so you will get this thing here, and this will bend over again. This is green, so this probably will go here, and then you will have again a synthesis, and then you will end up with 4 pieces of DNA instead of 2. So you start from 1, you went to 2, and you went to 4, and then you will go to 8, and so on, and you will get something like that. And this is called a cluster. It's a cluster of clones. In fact, it's exactly the same piece of DNA here, or perhaps complementary, doesn't really matter. And then what you do here then, since you have generated all that, you wash away whatever remains here, so this is clean, and then you start to synthesize each one of them in parallel. So it means that if you want to synthesize this one, let's zoom in a little bit. Okay, let's say that this is an A. Okay, let's make this smaller. A C C. What you do now is that you in the liquid mixture here, you put some special nucleotides that they can fluoresce different color depending on what they are. And not only that, but they have let's say a lag time. So if you put here a T with this fluorescent label, then you will not put another T immediately, but you have to wait a little bit. So what's happening is that a T will go here and a T will go here, and this emits, let's say, a red color, and the T will go here and the T will go everywhere simultaneously, so you will actually get a bright red color for all of these guys. Red, let's say. Now, I'm not sure if you will get from all of them, to be honest, because I see here that perhaps half of them are exactly the opposite, but they have found a way to do it properly. So from the molecules that you should get a T simultaneously, you will get a T, and probably it will be T here, and a T here, and a T here, and a T here, and a T here. You will get a bright red color simultaneously, and then you will proceed to the next one, which is again a T here. So again you will get a bright red color, and this process is described in this figure. So for example, you will get a T here, and then a G here, and then an A here from all the from all the molecules of DNA, from all the clones of DNA in the same cluster. So again, here is a cluster. It has clones of DNA exactly the same, which means that simultaneously all of them, they will emit the color that corresponds to T, which is red here. So this is red, and you will have a bright red color. This is G, and you will have a bright brown color. This is A, and you will have a bright green color. And then the machine is able to read accurately this red, and this brown, and this green, and so on. And eventually what you will get is the following. Okay, here they are. So eventually let's see, yeah. So what you will get eventually is these images, sequencing of such images, which means that in this cluster, so every spot here, this spot, okay, let's make it a bit larger maybe. Okay, this spot here, or this spot here, or this spot here, or these spots are different clusters of these clones of DNA. So exactly at moment 1, this cluster emits a green. So a green, if I remember well, was, let's say that green was G, which means that for this piece of DNA we have a G. Exactly at the next moment, this emits a purple, whatever it is here, and let's say that this yeah, I have the colors here. So okay, the green is a T. So the green is a T. Let's put it correctly. It's not a G. The green is a T, and then the purple is a G. So the next letter after T is G, and then it's red, which is a C, and so on. So this is the way that we obtain the sequence of DNA. Now the important thing, the parallelization here, is that you get this information for all clusters in parallel. So you know that this is T, for example, but you know that this piece of DNA, which is a different piece of DNA, has a T as well, and this has a T, and this has a C, and this has a C, and this has a, whatever it is, this is probably an A, this has an A, and so on. So instead of getting one piece of DNA each time and finding its sequence, you get the information for thousands of pieces of DNA simultaneously. And this is how we speed up the whole process. Now this is how Illumina works. I will say some details about how the data look like, and I will give some definitions a bit later.